I'm going to talk about a particular research study that we did entitled Fundamental Bounds on the Fidelity of Sensory Cortical Coding. And we're going to get into the details of this investigation shortly. But as a very brief introduction at the beginning, what I wanted to do was just to give the audience some background information about the general approach that my laboratory tends to take as we study phenomena in the brain. And our general interest from a biological perspective are questions concerning neural information processing and how the brain computes and transforms the data that it receives from many different sources, the outside world, inner world, and so forth. We often develop new optical approaches, optical microscopes, to address the questions that we have about neural computation. These novel technologies often produce very large data sets that can be first of a kind. And then we're faced with the challenge of having to analyze these large and novel data sets. They can often be in the several terabyte or even tens of terabyte size, which, you know, for perspective is say larger than the raw size of all the Hollywood videos that you might find on the Apple iStore. And the overall goal throughout all of this is to try to identify novel biological mechanisms that will help us to understand brain processing. So that's sort of our general approach. And today I'm going to try to illustrate this with a talk that focuses on a specific question, namely what limits the accuracy with which a large population of cells in the brain can encode a sensory scene. And the bulk of this was published earlier this year, a few months ago. I'm going to be focusing our discussion on the primary visual cortical circuits. And the talk is divided into 4 parts. So first, I'm going to introduce the question. And I'm also going to introduce longstanding hypotheses that have existed in the field since at least the mid to late 1980s, which posited that, in fact, it might be correlated noise between pairs or greater groups of neurons in the neocortex that may actually be the limiting factor on the accuracy of sensory cortical coding. Secondly, I'm going to discuss a new microscope that we built, as well as associated analytics that we developed to try to address our questions about the fidelity of sensory cortical coding. In the third part of the talk, I'll show you our biological results, and I'll try to explain what conceptual advance they allow for the field. Specifically, I'll present direct evidence for correlated fluctuations in large sets of neurons on the order of 1,000 cells or more. I'll argue that past measurements of mouse visual acuity are consistent with our measurements of noise in cortical processing. And I'll discuss how these measurements allow a new understanding of how the neocortex can compute accurately in the face of this type of noise. And then in the fourth portion of the talk, time permitting, I will discuss a number of new technologies that we've been working on that are coming over the horizon that will hopefully allow us to probe even further in future investigations the nature of brain circuit dynamics. And in particular, we're interested in how multiple brain areas work together in concert. Okay, so let's get started. So this is the first portion of the talk. So first, I'm going to give you some background information regarding the factors that are thought and have been hypothesized to constrain the fidelity of sensory cortical coding. Now, the general question of what might be the limiting factors on neural computation is actually a long-standing one. And luminaries in computer science, and in particular John von Neumann, one of the primary architects of the modern digital computer, considered this question intensively. And von Neumann, in fact, wrote an entire book on the topic and was very focused on the fact that the brain was not like a digital computer. It had aspects of both analog and digital computation in its workings. And therefore, that the accuracy limitations of neural computation were very likely to be different than those in the digital computer. Similarly, investigations with human subjects had revealed by as early as the World War II era that human subjects exhibited stochastic responses and stochastic perception to weak flashes of light, and moreover that humans can sense handfuls of photons. By the 1970s, the perceptual limits on weak sensory stimuli had been located in the primary sensory neurons, for example, in the retina, the photoreceptors, and also it was shown by Horace Barlow and colleagues, here's some example data, that retinal ganglion cells can respond to even a few photons. So there was the understanding fairly early on, decades ago, that for weak sensory stimuli, perceptual limits may be set, in fact, by primary sensory neurons and neurons just immediately downstream. But what about stimuli that are composed of greater numbers of photons? What sets the limit on the accuracy with which a sensory scene, a visual scene, can be encoded? So progress on this question came in the mid to late 1980s by Bill Newsom and his colleagues, Tony Mogshan. And this body of work was done in macaque monkeys, and the investigators used a set of stimuli which are now famous, known as the random dot kinetogram. And in essence, they showed monkeys sets of different moving dots, moving left, moving right, with different degrees of coherent motion between the dots. And what they found was that the extent to which the animal was able to discern the general pattern of motion to the left or right actually was pretty close in its sensitivity to the experimenter's ability to infer the direction of motion on the monitor, the screen that the animal is viewing, simply by listening to the spiking patterns of an individual neuron. And that latter threshold, that latter sensitivity, they called the neurometric threshold, as opposed to the psychometric threshold, which characterized the animal's sensitivity. So this was a very challenging result because it raised the question of why the animal's performance was not that much better than the performance that one would get in principle just by listening to the activity of a single neuron. So to address this paradox, Newsom, Mogshan, and colleagues proposed a potential explanation in the form of a correlated noise. Correlated fluctuations between pairs and groups of neurons might limit the ability of the brain to pool signals, average signals, across large groups of cells in an attempt to improve its estimation performance. So this slide shows you an example calculation of how signal to noise can be improved by pooling the signals of multiple neurons. You can see that the x-axis is a log scale. In the absence of correlated noise fluctuations, indicated by this curve here at r equals 0, correlation coefficient of 0, and then small to moderate amounts of correlated noise. And what these plots revealed is that even in the presence of a very small correlation in the noise fluctuations between pairs of cells, it can have a very large, there can be a very large effect at the ensemble level. So here you can see that in the presence of correlated noise, all these signal to noise ratio plots plateau and saturate. This occurs even for tiny amounts of correlated noise. And moreover, to be able to discern these effects accurately, one needs to be able to record from a very large number of neurons simultaneously, on the order of 1,000 or so. So this was a hypothesis that was very stimulating to the field and prompted a lot of work by theorists. And subsequent theoretical work showed that this line of thought generalized very nicely to ensembles of neurons with diverse sensory tuning properties. This particular calculation was done within a simplifying assumption that neurons would have comparable sensory response properties, but the general argument does not depend on that. And to give you a feel for how this works, many investigators have articulated and explored the fact that either positively or negatively correlated noise fluctuations can aid or impede the accuracy of sensory coding depending on the stimulus response properties of neurons in an ensemble or in a group. And to illustrate this point, I'm going to consider the world's simplest ensemble, as is often done. And these graphs here are based on a review almost 15 years ago now by Bruno Averbeck and colleagues. So here in this ensemble, we have only two hypothetical neurons. The response of neuron 1 is plotted on the x-axis, that of neuron 2 is plotted on the y-axis. And one should imagine that we are showing the subject, the animal subject, two different stimuli, and the task is to try to distinguish the two. Each point on these graphs represents the joint activity response of the two cells to either stimulus 1 or stimulus 2, which are denoted by the two different colors, the orange and the cyan. And then we can imagine further that the two neurons would either have positive noise correlations, negative noise correlations, or zero correlations, and moreover that the two cells could either have a similar response to the two stimuli, they might be similarly tuned, or they might be differently tuned. And what we find by looking at these different possibilities is that it's only the noise component that is parallel to the tuning curve or orthogonal to the decision boundary that affects the fidelity of coding. So in this case, the tuning curve, the direction that we care about in deciding whether stimulus 1 or stimulus 2 is being presented lies along this red dotted line. And you can see the schematized noise distributions. In the case of zero correlation, this decision becomes harder if there is, in this case, positively correlated noise that lies parallel to the tuning curve. But if the two neurons had negatively correlated fluctuations, this would actually aid the fidelity of coding. And we would come to an opposite conclusion if the two cells were differentially tuned to the stimulus. So the key point here is that it's only the noise component that is parallel to the tuning curve that ultimately affects the fidelity of coding. And this argument generalizes to larger populations of cells than just two. Now, throughout all of this, one important thing that had been missing was a direct experimental test of whether or not one could discern correlated noise of this type in large cell ensembles and actually pull out from data the kind of plateau that you're seeing here in this graph based on the hypothesis of correlated noise. So a definitive experimental test of these ideas had been wanting. And so this is where we put our attention. And in particular, we asked the question whether or not correlated neural noise might limit the fidelity of sensory cortical coding in the primary visual cortical area V1. So we went into the study with a number of different questions. First of all, we wanted to know whether one could measure this bound on the fidelity of encoded information by a neurocortical sensory ensemble. And if so, does that bound indeed arise from correlated neural noise of the type that I've been discussing? How many cells are needed to discern such an effect? At what population size does it rise? Is this noise actually limiting on perception? So is there any kind of correspondence that we could find between the bound on cortical coding accuracy and perceptual acuity? And then lastly, can we get any insight into von Neumann's question? How does the brain manage neural noise that is inherent to the stochastic dynamics of neurons to enable accurate computation? So this is something that doesn't arise so much in digital computation, in which the architecture of the computer largely presupposes faithful transmission of information, whereas in the brain, we know that spiking activity and many other processes are stochastic. How does the brain deal with that? And so we hope to be able to get some insight into that, that aspect of brain function from our investigations. And I'll come back to that point later. So to address these questions, we needed a combination of different methods that would allow us to directly observe ensemble neural noise patterns in a live animal, in the live neocortex. So first, because the scale of these effects is often at the, often arises at the size of ensembles of many hundreds or even 1,000 or more neurons, we wanted to be able to observe activity patterns of populations of cells of this order of magnitude of number. We wanted to be able to track the activity of V1 neurons across the retinotopic map, in other words, across the entire representation of the visual scene. We needed to have tools that would allow us to process the resulting terabyte-sized video data sets. and also analytic methods that were up to the job of assessing the accuracy of neural ensemble codes. And I will take you through all of these approaches, and I will show you that together they afforded us a new opportunity to measure the fidelity of neural population coding accuracy in V1, the primary visual cortex of the mouse. So now here in the second part of the talk, I'm going to introduce you a little bit to these techniques. So to image these simultaneous activity patterns of a couple thousand neurons, we designed and built a new form of two-photon fluorescence microscope. So this is a laser scanning microscope. The illumination source is an ultra-short pulsed infrared laser, the titanium sapphire laser. And I'm not going to go into all the details of this instrument. It was designed and built by Oleg Romyancev, Jerome Lecoq, a PhD student at Stanford and postdoc who focused a lot on the optical engineering and electronic engineering, and Joan Saval, a robotic and mechanical engineer. And together, they developed this instrument which divides the illumination beam from one laser into 16 different optical pathways. There are, in effect, 16 laser beams that are scanning the mouse brain simultaneously. Well, not perfectly simultaneously, as I'll mention in just a moment. And they together cover a field of view that is about 4 millimeters square in area, with a frame acquisition rate of up to about 20 hertz. And this can be done in the brains of awake behaving mice. So this was a large field of view that can be scanned at frame rates faster than was previously possible, and is enough to cover the majority of the mouse visual cortex, the primary visual cortex, I should say, V1. Now, I'm just going to highlight a few of the salient features of the microscope. So the division of the laser beam into 16 beamlets or different pathways allowed us to scan a greater area of the cortex per unit time than had been previously possible. And there was a corresponding set of 16 photomultiplier tubes, one for each tile of the 4x4 checkerboard pattern that we were using to interrogate the brain. And each of those patches had its own corresponding photodetector with which we could catch the fluorescence photons that were being emitted from the brain tissue. Now, I had mentioned earlier that it was very similar to having 16 laser beams scanning the tissue simultaneously. In reality, we were actually switching very rapidly at the scale of hundreds of nanoseconds between 4 sets of 4 beams that were activated. And I'll come back to this in a little bit. This allowed us to handle some of the effects of fluorescence photon scattering in the brain. Here you can see in the upper left in the panel that the different patterns of 4 beams that were illuminated at any instant, and we were using 3 electro-optic modulators to switch between the 4 different sets of 4. And this allowed us to improve the images in a way that I'll make clear in a moment. So in other words, each image pixel was cycled for only a quarter of the time. So you should imagine that there are 16 beams being swept, but in fact, only 4 of them are activated at any one of these phases of imaging. So we had phase 1, phase 2, phase 3, and phase 4. On the right, you can see the different tiles of the checkerboard that were activated in any one of these phases. And a key point is that throughout all of this, all 16 detectors were activated. And that means that we have the ability to observe and detect photons that might be generated, might be excited in one of the patches in one of these phases, but then would scatter in the tissue and be actually detected on a photodetector for a beam that was not activated at that instant. And that allowed us actually to computationally correct for some of the scattering of the fluorescence photons that occurs in the tissue. So I'm going to show you what this looks like. So the simplest way to piece together the image would be merely to add the images that one attained in the 4 different phases. And if you do that, you get an image. It looks like something here on the left. The scale bar shows you the size of one of these tiles, which is 500 microns. And perhaps you can see some of the seams between the tiles. This is due largely to fluorescence scattering between the tiles. But by using the information from all 16 detectors, you can actually correct for the scattering, unmix its effects, and arrive at images that look like those that's shown on the right here. And so this is an important part of the data processing that we perform. Now, we were using what's called neural calcium imaging to track the spiking activity of the neurons in review. And this is a common approach in the field. One can target a calcium-sensitive fluorescent indicator to the neurons of interest, in our case, V1 cortical pyramidal cells. And a large body of work in the field has shown that the modulation of calcium concentration as reported by these fluorescent indicators gives an indirect report on the somatic spiking activity. So one can think of it as an indirect readout of neuronal action potentials. And one of our control experiments illustrated here showed that after correction for the fluorescent scattering across the different tiles of our microscopes, there was almost no effect on our ability to read out the so-called calcium activity traces from the fluorescent signals that we were acquiring. And the way that we tested this, in essence, was to look at all the crosstalk from 15 tiles into one tile, and you can do that by turning off one beam and keeping the other 15 on, or you can do the reverse and have just the signal from one tile in the absence of crosstalk from the others. And by comparing these, we were able to show that the net effect of crosstalk was very small. Okay. One of the things we also took care to check was whether or not having all 16 pathways address the brain tissue might cause any discernible photo damage from the light or from the heat that was delivered. And so we conducted immunohistochemical studies to confirm that we did not see this under normal imaging conditions. You can see here some example data from the immunostaining experiments in which we looked for activation of a characteristic of a glial response. In this middle column, the GFAP is an astrocytic marker, as well as activation of the heat shock protein 70. We confirmed our ability to detect thermal damage in this way. That's the positive control. But under normal imaging conditions, in the bottom row, you can see that we see activation neither of the astrocytic marker, which would be an indication of tissue damage, nor of the heat shock protein, indicating that we see no signs of photo damage from the use of the microscope under normal imaging conditions. So to conduct these experiments, having built the microscope and validated its general use in brain tissue, we selected a transgenic animal that had been engineered at the Allen Institute by our colleague there, Honghui Zhang. And this was a triple transgenic mouse that selectively expressed one of these calcium indicators that I mentioned, in this case known as GCAMP6F, in a specific population of cells, the layer 2-3 cortical pyramidal cells. And so here you can see some example images from this animal, in which the fluorescently labeled cells here are colored green atop the black and white image that we got with this 16 channel 2-photon microscope. So now I'm going to show you what some of the example data look like using this transgenic mouse in our microscope. If you look carefully, you can probably just barely see the boundaries between the tiles of the 4x4 checkerboard pattern. You can see the blood vessels. Those are the larger stripes running through the image. And the little twinkles that you're seeing correspond to activations in the cell bodies of these layer 2-3 cortical pyramidal cells. And this is taken in the brain of an awake mouse viewing a monitor. Here's another example from another mouse. And in this mouse, you can see not only the somatic patterns of activation here in the cell bodies, but also you can see dendritic patterns of activation. And in this movie, we had improved our computational stitching of the different tiles, and they're probably harder for you to notice. Okay, now in this previous video, it may be hard for you to figure out or to deduce what the mouse is actually viewing on the monitor that it's seeing. But sometimes you can actually figure out what the animal is seeing. This is example data from a different brain area here in the colliculus, and we were actually showing a moving bar across the monitor, and you can see a corresponding moving bar moving across the visual representation here in the colliculus. So that gives you a feel for the data. So here in part 3 of the talk, I'm going to discuss how we brought these elements together to look at the bound on coding here in the sensory cortex. And so this was a collaboration between Oleg Romoyantsev, Jerome Lecoq, Honghui Zhang, as I mentioned, and also theoretical neuroscientist Suri Ganguly, who played an integral role, as I'll describe, in a lot of our analytic approaches. So this came out early this year, as I mentioned, and this slide introduces the visual stimuli that we used. So we showed the mice who were held in place under the 2-photon microscope and were allowed to view stimuli on a monitor that displayed either moving gratings at one of 2 different orientations, shown here indicated by stimulus A and stimulus B. You can see that the orientation of these moving gratings were generally plus or minus 30 degrees relative to the vertical. And they were presented in random order for 2 seconds in duration. And also these were interleaved by a 2-second interstimulus interval. And we monitored the activity of over 8,000 neurons in total from 5 mice. And the analysis that I'm going to show you, upcoming, focused on moments when the mouse's walking speed on a little treadmill that it's placed on was very low. And we did this because it has previously been shown that mouse locomotion alters visual processing. And we wanted to look at visual encoding under uniform conditions. And so we did this only at moments when the animal was stationary or nearly stationary. Okay. So the question now becomes, how well can one distinguish the 2 stimuli that we were presenting in random order on the monitor based on the evoked patterns of neural activity in visual cortical area V1? So that is going to give us insight into how accurately the cortex is representing them. Now this slide schematizes the computational pipeline that we use to process the resulting data sets. I'm not going to go through all the details of the processing, but simply to give you a flavor for how this works, there were generally 3 different stages of the analytics. The first should be thought of as pre-processing, and this takes care of a lot of the housekeeping operations having to do with a microscope. So we normalized the different gains from the 16 detectors. There were slight differences between them that needed to be accounted for. We did the unmixing to account for the crosstalk due to fluorescent scattering, as I was saying, registered the images over time to account for a little bit of brain motion that might occur. And then in the second stage, we could extract the individual neurons from these raw videos, as well as their corresponding calcium activity traces, giving us an indication of each cell's activity in response to the 2 stimuli that the mouse was viewing. And then we could begin in the third stage to analyze the activity traces of individual cells and of the population of cells. So first, we started off by looking at individual neurons, as well as the fluctuations that had occurred among pairs of neurons. And so we did this by looking at the so-called pairwise noise correlation coefficients. And we compared the coefficients computed for real data and also for what are called trial shuffle data sets, in which you would randomly permute the trials across different sets of cells. And so this is an interesting statistical manipulation because it preserves the distribution of each cell's individual responses. And I should say that we permuted this only amongst stimuli of a given type, but it breaks up the fluctuations that might occur trial to trial between different pairs of cells. And so what you can see here on the top left, first of all, for real data, is that these correlation coefficients that describe the strength of correlated fluctuations amongst pairs of cells are rather small, but nevertheless, they do seem tilted here in a positive sense. We also found a fairly broad distribution in the shuffle data, at least as compared to the real data. You can see that the variance is not that much diminished, but it's centered on 0. And this is interesting because it tells us that we are trial limited in the ability to measure pairwise noise correlation coefficients. So even in shuffle data, in which we seem to have attempted to scramble the data to remove such pairwise correlations, there still is a fairly broad distribution that is setting the limitations or telling us the limitations and how well we can accurately measure such pairwise noise correlation coefficients. Now, this is potentially important in the light of past measurements of pairwise noise correlation coefficients in the cortex. A number of groups have attempted to measure these pairwise noise correlation coefficients and have attained different answers depending on the preparation and the exact experimental conditions and the stimulus conditions. And this slide illustrates some of the difficulties in making accurate determinations of these pairwise values. Okay, so now we wanted to take a look at very similar issues, but at the scale of 1,000 neurons or so in these large ensembles, which we can uniquely interrogate with this new microscope. So the question then becomes, well, how do we actually assess in a rigorous mathematical manner the noise fluctuations in a population of n cells, where n might be something on the order of 1,000? So the issue here is that the noise covariance matrix has n squared elements. So in other words, if you have 1,000 cells, the noise covariance matrix would have nearly 1,000,000 elements. However, if we have only p trials in which we present the stimulus to the animal, with pn measurement data, we cannot accurately determine the noise covariance matrix elements if p is much less than n, which was the case in our experiments. So we could only have a couple hundred presentations of each stimulus. Nevertheless, to decode which stimulus was presented to the animal based on the responses of n neurons requires only n parameters, at least to specify a linear decoder. So perhaps from this perspective, pn measurements may be sufficient. And so I'd like to explore this point a little further with you. So with n cells, we can create a decoder that will give us an estimate of which stimulus was actually shown based on the recorded responses to that stimulus of the n cells under view. And with a linear decoding approach, one can do this by creating a hyperplane in the space of all the responses of the n cells. And again, I've tried to illustrate this point about neural population coding or ensemble coding using the world's simplest ensemble with only 2 cells. So here in this plot, responses of neuron 1 are shown on the x-axis and that of neuron 2 are shown on the y-axis. And then imagine that we're going to present 2 stimuli, A and B, again and again to the animal, and we're going to record the joint responses of the 2 cells, i.e. our ensembles. So each one of these data points represents a joint response of the 2 cells. And if we do that enough times, we can then catalog the distribution of points that represent the responses of the 2 cells to stimulus B and to stimulus A, and we can then draw in this case, a line that separates these 2 distributions, play the stimuli again to the animal, and then looking at which side of the line the response then falls and try to guess or estimate whether or not the animal was shown stimulus B or stimulus A. And so this would be an example of linear decoding, and it generalizes to populations that are larger than 2 cells. In that case, one tries to find an optimal hyperplane to make this classification between the 2 stimuli. Now, the way that we went about doing this was to perform a dimensional reduction. So we're working initially, as the data comes in something like 1,000 dimensional space with that many neurons. We used what's called a partial least squares analysis to find dimensions in that space that were informative about the classification, and then to build decoders in this space of reduced dimensionality. And so generally speaking, we worked actually in a 5-dimensional space in which we attained optimal decoding performance. And I'm going to show you a little bit what this looks like in a schematic that represents only 2 of these dimensions, only 2 of these informative dimensions from the PLS or partial least squared analysis. So now, as opposed to the prior plot, the 2 dimensions that I'm showing are those that are the products of the PLS analysis. So here you should imagine that at time equals 0, the stimulus has not yet been shown. One of the two stimuli can be presented on the monitor, and then the ensemble dynamics may take different paths as it develops, as the neurons respond. And for stimulus A, you can see here schematized a set of example trajectories in this space of reduced dimensionality, whereas these are some example trajectories of how the activity develops for the cases in which we presented stimulus B. So then the question becomes, how well can we distinguish the two stimuli based on these types of trajectories? So you can make a decoder, a linear decoder, as we did. This dotted green line is the schematized decision boundary. And the fidelity with which you can categorize the two stimuli can be assessed by this quantity d prime, a common metric in statistics, which is given by this factor delta mu, and that is the distance that separates these two distributions. So here, this red distribution and this blue distribution represent the projection of activity from these trajectories onto this optimal dimension. Delta mu basically tells you how far they are separated along this tuning curve dimension, and sigma here characterizes the width of these distributions. So you can think of this as a dimensionless separation between these two distributions along this tuning curve, and here we have the decision boundary. So you present the stimulus again, and you see on which side of the decision boundary the response lies, and you can characterize the stimulus in that way. So d prime tells us the fidelity with which that characterization can be performed. And in fact, d prime squared is a discrete analog of the amount of Fisher information that is encoded by the neural ensemble about the stimulus identity. So you can think of this as a way of assessing the information that's in the neural population response about the stimulus. So I'm going to show you some examples with real data of what this actually looks like. Again, using a space of only 2 dimensions. Here on the x-axis, we have the activity that's encoded within PLS dimension 1 and PLS dimension 2. And on the top row, we see this for real data. And here you can see the 2 different stimuli represented in blue and red for 2 different points, 2 different time points after the onset of the visual stimulus, at 0.15 seconds and at 1 second after stimulus onset. And indeed, here you can see some of the correlations in the noise. These are not spherical distributions. Whereas when we trial shuffle the data, we shuffle the responses amongst trials of a given stimulus type, you can see that they become much more circularly symmetric. So in this space of reduced dimensionality, we can create our linear decoders. This green line is the decision boundary for an optimal linear decoder that takes into account the shapes and the angles of these noise distributions, whereas this black decision boundary is that for the so-called diagonal decoder, which uses only the net displacement between the centroids of these distributions and does not account for the correlated noise fluctuations. And here in the trial shuffle data, you can see that the optimal decoder essentially corresponds to that of the diagonal decoder. Again, because in this case, we've removed the noise correlations. Now, a key facet of our ability to apply this analytic approach was the fact that we were able to show that we had sufficient numbers of trials to accurately compute these decoders as well as their corresponding d prime values. And so this was work done by our collaborator Surya Ganguly, who showed analytically and with simulations that the results from the PLS analysis could be accurate with far fewer presentations, far fewer trials than the number of neurons that we had recorded. And so these different plots show you the correlation between the actual and estimated quantities in a simulated model. And this general fact about the accuracy of our estimates held true for the eigenvalues of the noise covariance matrix, the estimated Fisher information, and were not true if we tried to estimate the individual matrix elements of the noise covariance matrix, as I had discussed earlier. And so this was a key piece of theory and simulation underpinning our approach. So now we can return to the data analysis and ask the question, how does D prime, the fidelity with which the neurons were encoding the stimulus identity, how does that scale with n, the number of neurons in the ensemble? And in particular, we wanted to be able to address the point that the older calculations had suggested, namely that the fidelity of coding should plateau with large numbers of cells. So on the previous plot that I had shown you, it had been signal to noise. D prime is very closely related to that. So we were looking for a plateau in the D prime values in the fidelity of coding with large numbers of cells. And moreover, we have the ability to perform a control analysis with the trial shuffle data, in which we have attempted to remove the correlated noise fluctuations between cells, and D prime should not plateau when we analyze those data sets. So here I'm going to show you the results from both the real and the trial shuffle data sets. You can see the real data in the green, the trial shuffle data sets in the orange. And here you can see the D prime values as a function of time since stimulus onset for these two. And when we examined subsets of neurons up to the size of the full number of neurons that we recorded. And we did this in two different ways. We did what we called instantaneous decoding, which we tried to classify the two stimuli based on the neural activity within a given time bin, or in what we called a cumulative manner, in which we used all the data detected up to a given point in time. You can see that for both decoding approaches, the instantaneous and the cumulative, that within about 0.5 seconds after the stimulus appears, the real data starts to plateau in its accuracy. You can say it takes a little bit longer here in the cumulative decoding approach. And in both approaches, we also see that there is a saturation as one increases the size of the cell ensemble. So after about 1,000 or so cells, one can no longer improve the D prime value. But this was not true, however, in the trial shuffle data as we had expected. So this was the first definitive evidence that there's a limit to how much one can improve the ability to discriminate between these two stimuli by adding more and more cells into the analysis. Now, we furthermore found that these diagonal decoders, which do not pay attention to or take account of the correlated noise among cells, actually performed only slightly worse than the optimal linear decoders. So here you can see that the asymptotic value of D prime with the largest number of cells was only slightly poorer using these diagonal decoders, meaning that at least for these two different stimuli that we were showing at plus or minus 30 degrees in orientation, it didn't seem like there was that much benefit in taking into account the shape of the noise distributions. I'll show you that this point has to be revisited when we use stimuli that are a little bit harder to discern. Another interesting point about this analysis is the number of cells that one would need to track to attain optimal decoding performance. And you can see it's on the order of about 1,000. Adding more cells beyond this, pooling the responses of more cells, does not lead to a substantial improvement in decoding beyond 1,000 cells or so. And so that's the scale of this effect. Now, as a control, we also used another analysis approach, regularized regression, to see whether or not it would give us different results than our partial least squares analysis. I'm not going to go through all the details from this, but here you can see a comparison of the results from the partial least squares analysis with our dimensional reduction to a regularized regression, which did not require dimensional reduction. And here you can see with this approach, we also get a saturation of the D prime value as we add greater numbers of neurons. So the results do not seem to depend on our exact analytic approach. Now, we also tried different visual stimuli that were closer to the acuity limits and harder for the animal to distinguish. So mice have rather poor visual acuity, and we presented in this second experiment visual stimuli that were oriented at plus or minus 6 degrees to the vertical. And these would be harder for the animal to discriminate. And in a corresponding way, we found that D prime values were were lower, as you would expect in the visual cortex. But again, we found a saturation of D prime for the real data, but not in the trial shuffle data. And moreover, we found that the diagonal decoders, whose performance is represented here with the black data, were now more noticeably diminished as compared to those of the real decoders, indicating that if you're trying to distinguish two stimuli that are rather similar to one another, that it probably does help to take into account the noise distribution when constructing the optimal decoder. And so this point is just stressed here in this slide. This was true for both the instantaneous and the cumulative decoding approaches. Okay, from these data, we were able to estimate what might be the minimum resolvable orientation difference that the mice could discern. So what might be the smallest angular difference that one might be able to distinguish based on the neural ensemble responses and the D prime values that we were getting. So an order of magnitude calculation estimates that it's about 2.5 degrees, which happens to be in quite close agreement with previous psychophysical or behavioral measurements of visual acuity in mice. So it seems that the behavioral measurements and what you would infer from the sensory cortical responses in V1 actually are in close correspondence. They're consistent. Finally, we took a look at what actually causes the saturation of D prime as we examine ensembles of cells of greater and greater number. And in particular, were there eigenvectors of the noise covariance matrix whose noise eigenvalues were growing with the number of cells, and in particular growing linearly with the number of cells, as would the visual signals. And so this would really, if we could find evidence for this, this would really show that the bound on the D prime values was in fact due to noise in the signal direction. So first, we took a look at the increase in visual signal with the number of neurons normalized by the number of cells. It grew linearly as one expected. And in real data, but not in trial shuffle data, we also found that there were eigenvalues of the noise covariance matrix that also continued to grow with the number of cells. And this is what would cause the saturation of D prime, but not in the shuffle data, as you can see. Now, we took a look in the individual mice at the top few eigenvalues of the noise covariance matrix. And here you can see there are in fact several that grow with the number of neurons. And this was typical across all the mice that we looked at. But not all of these noise eigenvectors will interfere with one's ability to discriminate between the two stimuli. And that is because, as I showed very early on in the talk, it is only noise in the tuning direction that actually matters in terms of determining our ability to discriminate the stimuli, that is actually information limiting, as it's called. So here you can see, for example, that in mouse 1, this first noise eigenvalue is much larger. This is the red one, the largest noise mode, is much larger, say, than this third one. But in fact, it actually seems to be the third noise mode that is information limiting, because here, this is the one that actually lies in the direction of the signal, in this direction delta mu, that is along the tuning curve. So what this implies is that there is an information limiting component to the noise, but it is not the largest noise mode. In this case, it was the third largest mode, and in some mice, it also was the second. Overall, about 90% of the noise that we found was in fact orthogonal to the direction in which visual signals were encoded, and thus this noise did not impede the representation of the stimuli. So to sum up what we found, at the level of pairs of cells, we found that there were small but measurable pairwise fluctuations in the cell responses. When one goes to populations of large numbers of cells, about 1,000 or so, we found direct evidence for correlated noise across these set of cells. Every mouse that we looked at had a few noise modes that grew linearly with the size of the neural ensemble, as did the visual signals. And the correlated noise, this correlated noise limited the coding accuracy. This correlated noise was important to take into account for stimuli that were very close to one another and hard to discern, but less important for stimuli that were not so similar. And moreover, the accuracy of cortical coding that you would infer based on the D prime values that we're assessing here in V1 are consistent with previous behavioral measurements of mouse visual acuity, including with these same kind of oriented gratings. And then finally, the interesting point that I mentioned at the end, namely that most of the noise is actually orthogonal to the encoded visual signals. And this point comes back to von Neumann's question about how the brain may compute accurately in the face of noise. And what this is telling us is that in many cases, the brain may not remove noise, but rather place it in dimensions that do not impede a particular calculation. So in this case, 90% of the noise is not problematic for discerning the two stimuli. Okay, so for this portion of the work, I'd like to thank a few more people that contributed. I mentioned many of the people that went along, but we had additional contributors, Yanping Zhang and Jane Lee and Rada Kopkevich, Oscar Hernandez, and this thanks also the funding sources that helped us. And I also want to point your attention to other recent experiments by the Averbeck, Harvey, and Marina Botte labs, which recently have found experimental results that are also consistent with those that I just shared with you. Now, in the fourth part of the talk, in the remaining few minutes that we have, I'm just going to introduce you briefly to some of the new technologies that we've been working on for giving us new ways of probing brain activity with optical methods. And one of the inspirations for this is that when we bring a new microscope or an optical approach to the study of brain activity, it gives us a new way of looking at very familiar phenomena. And I'm putting here this quote from Marcel Proust. Of course, he was not thinking of optical microscopes when he wrote this, but I think the message is very apropos, is that we can revisit a lot of the phenomena that are familiar in neuroscience, but if we have new optical technologies, we can see new facets of them from a neuronal perspective. So I'm going to share with you a number of technologies that my lab has developed over the years and recently. One that we're perhaps best known for is a miniature microscope that can be mounted on the head of a behaving mouse or another animal model. And this system is about 2 grams in mass. You can see it here shown on the tip of a finger. It carries its own LED illumination source and its own cell phone camera for recording neuronal activity. But unlike in the experiment that I just talked about, the animal can actually be freely behaving. And this system is based on semiconductor optoelectronics. And I do need to mention that this system has been commercialized, and I actually have a financial interest in the company Inscopics, which has done that, and we've distributed to over 400 labs now. There's over 1,000 microscopes in the field, and more than 360 papers have been published by groups using this technology. This slide shows you what it looks like in action. Here you have a number of mice wearing the head-mounted microscope. This was work done by Jones Parker, Biafra Hananu, and Jesse Marshall, and in this particular experiment, they were looking at the calcium activity of the so-called spiny projection neurons in the basal ganglia and dorsal striatum. And this gives you a sense of how we can simultaneously track what an animal is doing in an unconstrained way at the same time that we're tracking brain activity in at least one area. Now, a recent version of this system allows us also to manipulate neural activity in the field of view. We can simultaneously perform what's called optogenetics and activate or inhibit populations of cells in the field of view at the same time we're imaging neuronal responses. And that's a powerful combination of approaches. We've also been able to generalize the utilization of this approach beyond mice and rodents. Primarily in Japan, groups have been applying it to the study of freely moving monkeys, and in particular the marmoset, which has become a main model species that's used in the Japan Brain Minds project. Now, in some of the newer technologies that we have been creating in the lab, we have tried to broaden the perspective beyond the length scales that have been traditionally studied in neuroscience. So previously, there's been a lot of work at the synaptic level, at the cellular level, and focusing on simplified circuit models. What I talked about today was activity within microcircuits within an individual brain area. But one aspect of brain function which has received less attention is how different brain areas might work together. And so a lot of our technology development in the lab now focuses on multi-area activity and looking at brain areas working together in concert. So I'm going to show you a few approaches that we've been pursuing to do this. So one way is simply to try to image two or more areas concurrently. And we can do this with implanted microlenses that we can target to brain areas of our choice. And this takes advantage of the fact that even with a mouse brain, microlenses are sufficiently small that you can pack two or more into the limited area that you might have to target your selected brain areas. And so this was work that was initially done by Jerome Lecoq and Tony Kim in the lab. And one can do this in a variety of ways. This is data from Forey Wang and Tony Kim showing you that they were actually able to get two little small microscopes on a mouse. You can see here it's freely navigating a maze, and meanwhile these two systems are imaging activity in the prefrontal cortex as well as in the hippocampus. We can also do this using two-photon microscopy. This is a double-armed two-photon microscope that Jerome built. I won't go through all the details, but simply to say that each arm terminates in a microlens that can be mechanically moved and targeted to a brain area of your choice. And so one can target brain areas that might be nearby, for example, two different visual cortical areas in the mouse, or further apart. Here's a motor area and a somatosensory area that we imaged simultaneously. And because they're microlenses, you can even image deep areas. So here's an example of two simultaneously acquired images from the hippocampus and the frontal cortex of the mouse. Done in this case with a two-photon microscope, but analogous in some ways to what I showed you in the freely moving mouse. And I should mention that this is complementary to the approach that I showed in the first portion of my talk with a large field of view and the large microscope, and to that taken by other groups that are pursuing large microscopes and large cranial windows, because here we have a lot of mechanical flexibility to choose brain areas that might not be contiguous anatomically. So they don't have to be nearby on the brain surface. So I'm going to give you an example of this. This is work done in collaboration with my colleague at Stanford, Leach and Lowe, and spearheaded by Mark Wagner and Tony Kim. And here you can see a mouse that is performing an arm pulling task under the this double-headed two-photon microscope. And at the same time, we're able to image with one arm activity in the cerebellum, and with the other arm, we're able to image activity in the motor cortex. And so this way, we can watch what's happening in two cooperating areas of the brain that might be anatomically separated. Now, another approach we've taken is actually illustrated behind me in the background of the Zoom. We've been able to replace the natural cranium of the mouse with a curved glass window whose curvature matches the natural curvature of the cranium. And this can be done in a stable manner such that you can now view brain activity in an animal expressing an activity indicator. And this is an example actually in a head-fixed running mouse using the same kind of transgenic animal that I had described earlier. And those little flashing dots that you're seeing are in fact the firing patterns of individual neurons. And we can zoom in, and hopefully you can see a little bit more clearly now the activity patterns of individual cells, as well as some of the hemodynamics. And I'll zoom in yet again. And here at this greater magnification, you can see now, I think very clearly the activity of individual cells from this boxed area as shown here. And this is stable and allows us to come back repeatedly to large numbers of cells across a great number of brain areas, up to about 36 to 40. And we can apply different kinds of imaging, two-photon microscopy, one-photon microscopy to this, this crystal skull preparation, as we call it. Now, finally, I want to mention some approaches we've been pursuing about looking at neuronal transmembrane voltage directly. To this point, everything I've shown you has been indirect in the sense that it's used these genetically encoded fluorescent calcium indicators as an indirect readout of neuronal spiking. But we and others in the field have been trying to develop optical sensors of the neurons transmembrane voltage with a direct readout. So I'm not going to go into all the details of this, but this is an example of such an optical voltage indicator. This one was spearheaded by Yihang Gong when he was in the lab. He's now at Duke. And you can see a cell that is simultaneously tracked with an electrode. The electrical trace is shown here in black, and an optical trace from one of these voltage indicators is shown here in blue. And you can see the two are in close correspondence. And we can do this with only milliwatts of light. We've shown with control experiments in awake mice that we can catch every action potential from neurons. Here you can see a cell being recorded electrically at the same time that this green voltage indicator is giving us optical signals, and we can time the spikes to within about 200 microseconds spike timing precision. We can do this in different species, so I just showed you mouse. Chung Huang in the lab has shown we can do this with fly neurons. Here again, you can see concurrently recorded optical and electrical traces. And again, you can see interesting facets of the activity, these subthreshold depolarizations, and we can again time the spikes in the fly to 200 microseconds. Now we've been developing with colleagues, and in particular Vince Pirabone's group, Madhu Kannan and Ganesh Vasan at Yale, different colors of voltage indicators. These can be targeted to different neuron types. And in the fly, Chung has shown we can actually track these different neuron types over long periods of time in a way that's analogous to what I showed you with the this clear glass cranial replacement in the mouse. And to give you a sense of what this looks like, here we can come back to individual neurons again and again over multiple days with these voltage indicators, imaged at a kilohertz, and track individual spikes. And in fact, one can do this in 2 different types of neurons at a time, which we hope will allow us to dissect the means of cooperation between different types of neurons. So to give you a closing movie of what this looks like, here you can see a head fixed but behaving fly. And meanwhile, we are using optical voltage imaging to track these spiking patterns in different sets of neurons concurrently. So that's a taste of some of the technologies that we've been working on. Just to sum those up, we've been using micro-optics, as you saw with the miniature microscopes and with the imaging of deep areas. This can be done in freely behaving animals, and particularly in Japan, groups have been doing this in primates. We've been trying to move into voltage imaging, which gives us single spike resolution at very high time resolution as well. And one can also potentially look at voltage rhythms. And I showed you a few of the approaches we've been using to look at more than one brain area at a time, targeting 2 or more brain areas concurrently, or looking at very large fields of view through these curved cranial windows. And so with that, I will stop, thank additional funding sources and collaborators, and see if there are any questions to be addressed. Thank you.